Alright, let's talk a little bit about the viable count assay. Now, this is a very important technique when you are in a microbiology lab. And you have a sample, and that sample can be anything. It can be a food sample, it can be a soil sample, anything. And you want to calculate the bacterial load of this sample. So in that case, what do? You homogenize the sample, and in 1 ml of that sample, you calculate how many units are there which can actually form a colony. So the whole thing is colony forming units per ml or per ml of the solution. Right. So let's start to calculate. So first we have a sample, and from that sample, like I said, you homogenize the sample and you take 1 ml of that sample and you dissolve it and mix it in a test tube which is having 9 ml of distilled water. Right. So 1 ml is going into 9 ml, and the final volume, it is going to be 10 ml because both of them are liquid. Now what do? You take out 1 ml from this, or 0.1 ml from this, and you put it in another test tube which is having 9.9 ml of double distilled water. Right. So this is basically you are serially diluting your initial sample, right? So that you can get an idea of of how many bacteria could be there in a diluted sample. Now you again take out 1 ml from this second dilution and you put it into 10 ml of water or double distilled water. Right. Now again you take out 0.1 ml and you put it into 9.9 ml of double distilled water. Right. Finally, let's do it once more. Take out 1 ml and you put it into 9 ml of double distilled water. So these are the 5 dilutions that you make, and you serially diluted the sample. And now what you need to do, you need to calculate the initial dilution of the sample. Right. So the initial dilution, the formula is stock volume by the final volume. Right. Stock by the final volume. So for the first one, we took 1 ml from the stock and we are diluting it into a final volume of 10 ml. Why 10? Because 9 and 1, like I said, both of them are liquid, and so the final volume is going to be 10 ml. So 1 upon 10. Second one, 0.1. Now this is a stock and we are diluting it again in final volume of 10 ml. Similarly, it is again 1 by 10. It is 0.1 by 10. Finally, 1 upon 10. So these are my dilution. Let's simplify them. The first one would be 10 to the power minus 1. The second one would be 10 to the power minus 2. Third one, again 10 to the power minus 1, 10 to the power minus 2, and 10 to the power minus 1. So these are my 5 initial dilutions. Now what do? You sort of take out the final dilution of this of these 5 dilutions. So for the final dilution, the first one would be 10 to the power minus 1 because it is the initial dilution and this is from where we are starting. So this is the same as the initial dilution. Now for the second one, the final dilution would be, since you are taking this, since you're taking the second one from the first one and then you're diluting it. So you're taking it from this sample and then you're diluting it. So the final dilution would be different, and this would be, you need to multiply the initial dilution of the second one with the final dilution of the first one. And what do you get? You get 10 to the power minus 3. Right? Similarly, you need to multiply this to this, and the final dilution you will get 10 to the power minus 4. Again, for the fourth one, 10 to the power minus 6, and finally 10 to the power minus 7. Right? So now you have the final dilutions calculated. So next what you do, you take 1 mils from each of the dilutions that you make. You take 1 mils from each of the dilution, 1 mils, and you plate it into nutrient medium, and you let the bacteria grow. Right? So you plate it nutrient medium and you let the bacteria grow. After growing, you calculate the number of colonies. So next step, number of colonies. You need to calculate that in each plate. So for the first dilution, you saw the number of colonies that were found were 1000. For the second one, you got the number of colonies to be 500, then 250, then 10, and the final one had no colonies. Right? So these are the different dilutions. These are the different dilutions that were plated and the number of colonies that were obtained. Now you can calculate the CFU per mils, or the viable count. So for that, CFU per mils is equal to number of colonies divided by the volume or the amount plated, the volume plated, multiplied by the dilution factor. Mind it, this is the dilution factor, but till now we were talking about only the dilution, the final dilution and the initial dilution. We did not talk about the dilution factor yet. So dilution factor is nothing. All you need to do is let's do it from the beginning. So you have number of colonies 250. Alright, so I need to tell you why I'm taking 250. Alright, so we need to consider a plate which has the number of colonies which is less than 300 and greater than 30. Right? Why is it so? Because if it is greater than 300, we consider that to be too numerous to count, or TNTC. And if it is below 30, we call it too few to count. Right? So 500 over here, this is TNTC because it is greater than 300, and again 10 over here, it is TFTC because it is too few to count, it is less than 30. So the plausible plate that we can take is this 250, and that is why the number of colonies in the calculation, we are going to take it to be 250 divided by the volume plated. We plated 1 mils of that dilution, so 1 multiply the dilution factor. Now what is the dilution factor? Dilution factor is nothing but the reciprocal of the final dilution of the plate that we are actually calculating. So for the 250, we have final dilution of 10 to the power minus 4. So the dilution factor would be 1 by 10 to the power minus 4, or just reversing of the sign, that is 10 to the power 4. So this is the dilution factor. Now if we calculate it, 25 multiplied by 10 to the power 5. So this is the final answer, which is the CFU per mils. That is, these many number of colony forming units are present in 1 ml of the sample that we plated. Right. So this is the final answer, and this document I'm going to be linking it down in the description, and you'll find a Google link to that, Google Drive link to that. You can download it for free and you can refer to it when you're looking at the video. And that's about it. I'll see you in the next one. Bye-bye.